Yes, so thank you everyone for joining. I was just saying that this is actually the highest number of people we've had at a TSA event, virtually, that is. So it's absolutely wonderful to see everyone. And it's just a shame that it's in such trying times. Genuinely, for myself, so I'm Luke, I'm the head of communications and support at the TSA. I do hope that everyone's doing as well as can be expected at this time with everything going on. So a quick recap as to what this event is all about. So we had the TSC Research Symposium event last Friday, which was focused on professionals, TSC professionals and clinicians, talking about the latest research in TSC as a whole. And we were really keen to ensure that everyone in the community had an opportunity to hear about the latest research as well. So we're absolutely delighted to have Dr Frances Elmslie and also Professor Julian Sampson with us to give us an overview exactly on that, to see what the latest findings and whatnot in TSC research is. There will be opportunities for questions as well. If you do have any questions, please just put them in the chat box on Zoom, and we'll be monitoring them. In the event that we do run out of time for questions, we'll see if we can answer them after the event, as and where possible. So yeah, without further ado, I will introduce you to, as I say, Dr Elmslie and Professor Sampson.
Okay, so I'm Frances Elmslie. I'm a clinical geneticist at St George's, and I run a tuberous sclerosis clinic. And I'm also a trustee of the TSA. And I'm really delighted to be chairing this session for you today, which, as Luke has said, is a summary really of the research symposium on the 13th of November. And I'm really grateful to Professor Sampson, because actually he's done all the hard work really. So he's going to be presenting the research, and I will then hopefully just have a look at the chat, look at the questions, and try to either answer them myself or ask Professor Sampson to answer them. I'm sure Professor Sampson will need no introduction to any of you. He's worked on tuberous sclerosis for a lot longer than me, and was part of the group that identified TSC2 back in 1993. So it had a very long-standing research interest in TSC. So what we've decided to do is to actually condense the talks down into some themes rather than presenting them as the individual talks. So we're going to start off with talks on TSC and COVID-19, and then go on to talk on LAM, one on the renal cystic disease in tuberous sclerosis, and then we're going to finish up with the 3 talks on TAND and autism. So, Julian, I wonder if I can hand over to you to share your screen and kick off with TSC and COVID-19.
Thanks, Frances. So, yeah, I'll try and share the screen in a moment. And if there's any problems, I think because there's just a few slides to illustrate main sort of topics. And if there's any problems, I think Luke's got them and he can bring them up if my screen doesn't share. But anyway, good evening, everybody. And I hope we're going to have an interesting hour listening to and discussing the evidence and talks that were presented at the TS Research Symposium. So what I've tried to, I don't know, it's possible, I guess, that some people on this call actually already have listened to the whole symposium or have had access to some of the online things from it. If so, apologies if I've oversimplified anything as I talk through it. I think, as Frances said, if you've got some questions, if you can use that chat bar facility in the sidebar to just put any questions down, Frances will sort of pull those questions together. And then what we'll do, Frances, is after each of these sort of topics, the 4 topics, we can stop and have questions before we go on to the next one, just so that we keep, we keep things as simple as possible. So I'm going to try and share my screen. We shall see if it works. Oh, it looks like it's going to, remarkably enough. Let's see how that goes. Can people see that?
Yeah, we've got that.
Yeah, fantastic. I'll just have to move some faces out of the way here. So that's okay. So yeah, we had a really interesting day on the Friday, looking at a number of aspects of TS research. And of course, one of the most sort of hot topics, but it's still early days, was what COVID-19 meant really for people affected by tuberous sclerosis or sporadic LAM or tuberous sclerosis with LAM. And we had two international speakers who were able to tell us about their experience and their research. And they were Dr. Angela Peron, who's a medical geneticist from Milan in Italy, and Dr. Nishant Gupta giving a talk. He's a lung specialist based in Cincinnati, which of course is a major center for TS research internationally. So it was great to have these two international speakers. And the first one to tell us about her experience was Angela. Working, of course, in Milan, she was right at that sort of that focal point of the first wave of infection with COVID-19 in Europe. We'll have to go down here to move on slides by the looks of things. And so she told us about basically their experience in Milan during the period from the beginning of January through to the end of April. And then she updated it with a little bit of information about what had their experience that they'd had between April and October. In fact, they'd already published, they've just published a paper describing their experience of COVID in the TS patient group during that January to April period. And so that can actually be found by simply Googling TS Milan and COVID, and you'll find a link through to that paper, which is freely accessible to everybody. So what Angela told us was that they had they basically collated their information from all the patients that they'd either seen in their TS clinic or had virtual consultations with in place of the TS clinic during these months of the first wave. And that included 93 patients with tuberous sclerosis, another 9 patients with sporadic LAM. And I think about 13 or so of the patients with tuberous sclerosis also had significant LAM. So they had 20 odd patients with LAM altogether. And if we look at the 93 plus 9 patients altogether, 102, 30 of those were children and 72 were adults. And I think the youngest child that they'd been following up during this time was only a few months old, and the oldest adult was 72 years of age. So they had a very representative spectrum of people with TS or sporadic LAM. And of those 102 patients, 26 were taking mTORI inhibitors. So that's sirolimus or everolimus. Everolimus, some of you may also know as Vertubia, because these drugs unfortunately often have 2 different names. So what they had done in Milan is they had recommended to all their patients who were on an mTOR inhibitor that their patients should continue the mTOR inhibitor, even though the pandemic was getting a real grip on northern Italy. 2 patients out of the 26 actually stopped taking their mTOR inhibitor when they developed symptoms suggestive of COVID-19, but I believe that in fact, in those 2 patients, the symptoms proved not to be COVID-19 in the end, but just another virus. So this advice that they gave was a little bit different to the advice of some centres. I think at the beginning of this, different TS clinics were giving different advice to their patients. Really, we had no experience to go on. I know here in Wales, we reviewed the position for all of our patients who are on mTOR inhibitors and made sort of individual recommendations to families, which they could then follow or ignore, of course, depending on whether they liked our advice. But in some cases, we felt that it was important that the mTOR inhibitors were continued, for example, in patients with who had rapidly progressive LAM. And in some patients, we recommended reducing the dose, and in other patients, we recommended actually stopping for a period while we saw what happened with the COVID pandemic. But in Italy, they just recommended to their patients that they continue with their mTOR inhibitor treatment. So by October, what Angela said was 7 out of the 102 patients had definite COVID-19. In 5 of those cases, antibody tests had shown that the patients had COVID-19, and a couple of them had COVID-19 confirmed by swab tests. So these are rather different tests, one detecting the virus and the other one detecting the antibody after infection. And they found that 2 of the 7 had no symptoms at all, 3 had mild fever, 1 patient had typical symptoms, and sadly 1 patient, a 61-year-old male who had severe tuberous sclerosis, had died following admission to the intensive care unit. That patient was not taking an mTOR inhibitor. What they felt was actually this was pretty representative of what was going on in their local population in the Milan area at the time. So it didn't seem, they had 26 patients taking an mTOR inhibitor. It didn't seem that the infection had been more prevalent or more severe in patients taking an mTOR inhibitor. So I think that was some useful information based on this experience in Milan. And in fact, Angela had some thoughts about this. She noted that in fact there had been previous interest before the pandemic in the possibility that mTOR inhibitors might actually work as treatments for some viral infections. And this had been postulated because mTOR inhibitors block a lot of functions in the cell, including protein synthesis. So there was some evidence that mTOR inhibitors like Vortubia and like sirolimus actually might block viral replication. And perhaps more surprisingly, there was also some evidence that mTOR inhibitors may affect the immune system in a way that actually makes the cytokine, so-called cytokine storm, this is the severe disturbance of the immune system that occurs in severe COVID-19 infection. So some evidence that mTOR inhibitors might actually damp down that cytokine storm and therefore have the potential to prevent the most severe COVID infections. But this is really a bit of speculation, I think, rather than fact at this point. So I felt that was pretty reassuring that people with tuberous sclerosis and people on mTOR inhibitors don't seem to be particularly more vulnerable than one would otherwise expect to COVID infection. The second talk in this area was from Dr. Gupta, the lung specialist from Cincinnati. And he reported some quite complicated science about preclinical experiments, so experiments in cells and mice. And many of these related to the use of mTOR inhibitors in viral infections. So the first thing he noticed was that not COVID, but flu viruses in mice in the laboratory can kill laboratory mice. And in fact, mTOR inhibitors were found to reduce deaths from the flu virus in mice. He then reported on a small study that had been undertaken in China in patients in ITU, again with flu infections, not COVID infections. And that study had suggested that mTOR inhibitors actually reduced the death rate in patients in ITU who were being ventilated because of severe flu. So that was one in an animal model and one in human, 2 studies that suggest that mTOR inhibitors might actually be beneficial in some viral infections. He then cited some further evidence from a study of people, elderly people over 65 years of age, who were having flu vaccination and looking at the antibody levels that the vaccination led to. Basically, it's thought that a high antibody level after a flu vaccine is a good thing. And what was found that actually small doses of mTOR inhibitors given alongside at the same time as the immunization seemed to lead to actually higher antibody levels. So again, suggesting that mTOR inhibitors might have some beneficial effects in the immune system. And on the basis of this, Dr. Gupta has actually started a trial of sirolimus, that is rapamycin, in COVID patients in Cincinnati. This isn't COVID patients with TS, this is just COVID patients who are in hospital with severe COVID. Now, we don't have the results of his trial as yet. I don't think it's fully recruited, but I felt that from these first two talks, there were some conclusions that could be made. First, that although there's little data so far on tuberous sclerosis, mTOR inhibitors and COVID, what data there is quite encouraging. I would still suggest that people affected by tuberous sclerosis and people taking mTOR inhibitors are cautious and do everything that they possibly can to avoid contracting COVID-19. But if people with tuberous sclerosis or mTOR inhibitors do develop the condition, I think the data so far suggests that, that they will not fare any worse than anyone else. So mTOR inhibitors in TSC don't seem to be associated with major COVID problems or unexpected COVID problems. And mTOR inhibitors might actually have some beneficial impact on COVID infection. And hopefully Dr. Gupta's trial, when it's completed in Cincinnati, will tell us whether that is the case or not. So, so that was the first talks about COVID. And I wondered whether there were any questions for either myself or for Frances. And I'm just working out how to get back out of the screen sharing. There we are, so that you can see us again. Sorry, there we go. So, Frances, has anything come through?
Yes, so just one question so far, which I think we slightly anticipated, is somebody's asking, I think everyone will be wanting to know, can TSC patients take the C19 vaccine safely? Do you want to have a crack at that one?
I can have a crack at it. So I think the answer to that question will be yes, that patients with tuberous sclerosis should have the vaccine like anybody else. What we don't know at the moment is what the criteria are going to be for speed of access to the vaccines. So I would say, that at the moment, it looks like our government in the UK is wanting to base this mainly on age. And I know that governments in other administrations are wanting to take into account underlying illness as well, which is much more complicated to do as a policy for immunising the whole population eventually. So I rather suspect it will be on the basis of age. And so people who are young may not get access to a vaccine quickly. But if you do, I would definitely recommend taking it. And I certainly will be.
Yes, me too. So just on that theme, and don't worry, Mary, I will come back to your question, but carrying on the theme of vaccine, Marie James has asked about whether mTOR inhibitors, because they block viral replication, how that might affect a potential COVID vaccine.
Yeah, so a very good question from Marie. Because there are different types of vaccines. It looks like most people in the UK are probably going to get the AstraZeneca vaccine, which is quite a conventional one, in which the protein, the spike protein, is really what's used in the vaccine. And the evidence that we cited from the flu vaccine example, which is a similar type of vaccine to the AstraZeneca Oxford coronavirus vaccine, suggested that actually, certainly in older people, that an mTOR inhibitor actually helped with the immune response, which is surprising and a bit counterintuitive, but interesting. So I think that for that type of vaccine, I can't foresee any reason to suspect a problem if you're taking an mTOR inhibitor. The other vaccines that have shown promise recently, they're the so-called RNA vaccines. And in that situation, the RNA is used by the cell machinery in the body to make the protein. And in theory, an mTOR inhibitor damps down protein synthesis. So you might think, oh, well, that's not going to be such a good thing. But I suspect that any effects will be so marginal that, that they're unlikely to impact significantly on the efficacy of the vaccine. But clearly there won't be any data on this when the vaccines become available. So I think, my recommendation would be to carry on with the mTOR inhibitor therapy if you're on it and have the vaccine.
Okay, I think you very neatly answered 3 more of the questions about the vaccine. So thank you very much for that, Julian. And so Mary had a question about the age of the 2 asymptomatic cases. I think that was in the Italian, obviously in the Italian study. My memory was that we didn't have the specific ages for those 2 cases. I think it was just a summary. Have I remembered that right, Julian?
I think you're right. We could go back to the original paper, or in fact, Mary could access the original paper and it's got the details on it. I was looking at it earlier today, but I honestly can't remember for those asymptomatic cases, because this was really later on. So it may not actually be, that data may not fully be in the paper, because what Angela told us about was, first of all, what she'd said in the paper, and then told us about the information that had emerged in subsequent months, which isn't yet published. So I suspect we just don't know. On the whole, but I know some of them were children.
So, yes, so it's possible
they could have been quite young.
Yeah, okay. Yeah. And Adrian has asked a more specific question about a 7-year-old who started everolimus for kidney AMLs, but was then stopped for 5 weeks due to COVID. Should we push for him to start this again? So all I can tell you is that in our practice in St George's, we have restarted everolimus in the vast majority of patients, and in those that haven't restarted have done so for personal reasons. So from a health professional perspective, we have encouraged people to restart. And would you say the same in Cardiff, Julian?
Yeah, I think we would, particularly given the emerging data about the mTOR inhibitors being quite safe. And it's a balance of risks, isn't it? So, I guess that the 7-year-old patient in this case must have quite large angiomyolipomas to be on the treatment. So I think it would be important to keep those under control, because we know that stopping everolimus will lead angiomyolipomas to regrow. And increase the risk of bleeding. So I would absolutely say that. I don't know, Frances, what your view would be. Of course, if a 7-year-old catches COVID, they're very unlikely actually to develop any symptoms at all. But if you did have a patient on an mTOR inhibitor who had symptomatic COVID, would you still be happy for that person to carry on, for that child to carry on treatment, or would you just stop it for a few days until the infection had passed?
Yes, so that has been the plan. We actually haven't had any patients. We have about 60 patients on mTOR inhibitors, and none of them, to our knowledge, have had COVID, though of course many of them could have had it asymptomatically, I suppose. But that was going to be the plan, that if people did catch COVID, we would recommend just stopping it while they were actually symptomatic. Probably, I don't think we specify, but probably for a week or so. And that's, we know that stopping for a week would not be associated with any adverse effects. Just to say, exactly like you, we decided to keep the people with LAM and deteriorating lung function on everolimus or sirolimus, on an mTOR inhibitor, and not stop it. Whereas actually we took everybody else off, although some people chose to stay on their everolimus treatment, but we kept them off for 3 months and then they restarted subsequently. I thought I should also just add here, actually, that I forgot to mention right at the very beginning, we had a lovely talk from a patient who had sporadic LAM, not TSC associated LAM, just talking about her experience, because she developed COVID, and actually she sort of rode the storm, as it were, very, very well. And so although she does require oxygen, at times for exercise. She's not on oxygen all the time, but she is, from memory, she was taking an mTOR inhibitor, and actually she saw her way through COVID with no adverse effects. So that was obviously one individual, but just worth mentioning. So we seem to have come to the end of the chat, or end of the questions. So shall we move on to the next talk, which continues our theme actually quite nicely, because this is the talk from Simon Johnson about LAM.
Okay, so there we are. I'll just go into this. So yes, so Simon Johnson, some people may know already. So Simon's from Nottingham, where he's a doctor and a researcher who specialises in LAM. He runs the National Centre for LAM in Nottingham and has undertaken much research into this condition. And his talk, again, was quite a detailed scientific talk, but he was looking at an area that was new, certainly for me, and that was the role of so-called mast cells in LAM. So mast cells, we've all got them, and these are immune cells that mediate an inflammatory response. So typically are involved in allergic-y type reactions and inflammation in response to an antigen or something that provokes the immune system. And what Simon and his team described was that they had developed a culture system where they had cells from LAM nodules, and these were human LAM nodules, and non-LAM cells that they were able to culture together. And he was also using a mouse model, so a mouse in which LAM cells were injected and travelled to the lung, creating the mouse equivalent of LAM nodules. And then he was able to study both the cell culture system and the mice. And what he basically found was that the mast cells seem to be attracted to LAM nodules. These are migratory cells that move around the body. They move into the LAM nodules, and they release chemicals in those nodules which seem to contribute to lung damage. So there are many different factors that lead to lung damage in LAM, but this seemed to be one of them, or certainly potentially. So based on both the cell culture models, and then he'd actually gone into the tissue bank of LAM tissues from patients that are kept in Nottingham and looked at the mast cells and found evidence of activation of these mast cells in these stored lung tissue samples as well. And what they found was that they could actually block this mast cell activation using a drug that's commonly prescribed for asthma called sodium chromoglycate. These used to be called, I think they probably still are, Intel inhalers. So really a very safe drug. And also that this response was blocked by the mTOR inhibitors. So I think what this research from Simon seemed to show was that this is yet another facet to the very complex pathology of LAM, but is one in which these treatments offer some potential advantage. And so I expect we're going to hear more about treatments that target mast cells in LAM in the future. So I think that was all I was going to say about Simon's talk, because a lot of it was quite complicated stuff about cells and mice rather than patients. But again, any questions in relation to that research or LAM, we could probably take them now.
Yeah. So none in the chat just yet, but Philip has a very sensible question alluding to the previous discussion about whether to stop everolimus if somebody had COVID. And this is with reference to his son, who's in a residential care home with 8 other residents. Would we stop giving everolimus or sirolimus in the event that a fellow resident contracts COVID? So to be honest, this is not a situation that we've actively discussed in our team, but my own view would be that these are young people, and so actually they are generally low risk, and if anything we're showing that mTOR inhibitors may be to some extent protective. So I wouldn't stop giving it if he is asymptomatic. That would be my own view. Julian, would you agree with that?
I would now. This question did come up way back when, in sort of February or something, when the pandemic was just getting going and was clearly on its way. And I did discuss this with a number of parents of younger adults who were in care homes. And at that point, we were feeling a bit more worried about it. And I actually said, well, perhaps, if COVID gets into the care home, then stop the mTOR inhibitor because of worry about its immunosuppressive or potential immunosuppressive properties. But I think, because of what we've learned through experience over the course of this year, I now would say, no, don't stop it. Don't stop it if someone's asymptomatic, because I think the consequences of stopping are likely to be worse than of continuing. And particularly, if a care home has a problem with COVID, then it's not going to just be one person who'll catch it. So, you potentially be stopping for quite a long period of time until the infection was cleared from the care home. So I'd agree. I think, keep going.
Yeah. So another question. This is about LAM. So this may appear naive. I don't think it does at all, actually. But would therefore an inhaler help with treatment of LAM if a previous drug is deemed safe? So you're obviously referring to the sodium chromoglycate, and that is indeed a suggestion. So I think I think there are plans, are there not, to do a clinical trial of this? Or I now can't remember. I think I asked a question about this actually in the symposium. Is Simon planning a clinical trial, or they're certainly considering the use of sodium?
Yeah, I thought they were making these plans because I think the evidence in terms of using the models, I think Simon had shown evidence that if you treated early with sodium chromoglycate, that this could reduce the damage that was occurring in the model system. And sodium chromoglycate is a very safe drug indeed. So yeah, I think there would be, I think that is their intention, and it seems very sensible to look at this in a clinical trial setting. And absolutely. People are even looking at using mTOR inhibitors through an inhaled route. So using an inhaler is a very sensible way of getting things into the lung, if the lung is where you want it. Yeah,
absolutely. So that's got a wow, which does seem absolutely beautiful, I think. So let's hope. So we're getting amazing, great news. So let's hope that this does live up to its promise. But I agree. I think it sounds really interesting and potentially really exciting for the management of LAM.
Yeah, and of course this is work that the TSA has been funding, and supporting. So so yeah, I think it's a good outcome so far. But I think for patients affected by LAM, then we really have to wait for the outcome of clinical trials. There's so many treatments that look good in cell models or in mouse models that don't make it when you try and then use it in the real world. So there's a big dropout. So I'm glad people are excited by the prospects, but don't get too excited until the clinical trial data are published.
Yes, absolutely. Always a note of caution. Great, good. So let's move on to Professor Bissler's talk on renal cystic disease, which I have to say, for me, I found extremely educational, because I hadn't quite appreciated the real range of renal cystic disease that one sees in tuberous sclerosis from a non-nephrologist's perspective. So carry on, Julian.
Yeah, okay, let's bring this one back now. So yeah, John Bissler, for those who don't know John, is a pediatric kidney specialist who worked for many years in the Cincinnati tuberous sclerosis clinic, and then moved and set up his own clinic in Tennessee, in Memphis, which is now a fantastic multidisciplinary clinic led by John. And he's had a huge amount of experience over many years of kidney problems in tuberous sclerosis. And historically, most of his work has actually been on renal angiomyolipomas, but he's also very interested in kidney cysts in tuberous sclerosis and has become a bit of an expert in this. And he feels that there are a number of different types of kidney cyst occurrences in tuberous sclerosis with different causes and different outlooks, different chances of causing problems with kidney function. And I certainly agree with him that there are different types of kidney cystic disease in tuberous sclerosis. But I think what John's done, being a real specialist in this area, is he's really divided these up into into these discrete types of cystic kidney disease in a very expert way. So he defined these 5 different types of cystic disease in tuberous sclerosis. So that's 5 so far. I expect when we hear from him in a few years' time, there'll probably be even more. The first is the one that is perhaps best described in terms of its genetic causes, and that is the TSC2 PKD1 deletion, where the tuberous sclerosis gene on chromosome 16 TSC2 and the PKD1 gene that causes normally adult or autosomal dominant polycystic kidney disease, that it's located right next to TSC2, and so these 2 genes are often damaged together. And where they are, the person develops lots of kidney cysts, often in childhood, sometimes even before birth. And this can be pretty serious and can lead to kidney failure requiring kidney transplantation or dialysis in early adult life or later on in adult life. So this is a pretty severe form of cystic kidney disease. And I think, usually when we look at the scans of the kidneys, we can take a pretty good guess that someone will have this type if it's present. The second type that John described were cortical cysts, and that means sort of out nearer to the edge, nearer to the outside of the kidney. And these cysts are usually smaller, they're usually less in number, and they're very, very common in people with tuberous sclerosis, but they don't usually lead to kidney failure. And so these sort of cysts, I think, are much less worrying and mustn't be confused with the more severe forms of cystic kidney disease in tuberous sclerosis. And then the third type he described was what he called TSC multicystic disease. And this was a form of cystic kidney change where the cysts were present throughout the kidneys, both in the sort of inner part of the kidney and in the outer parts of the kidney. But unlike the TSC2 PKD1 form, this only rarely seemed to lead to kidney failure and could be seen in patients with either TSC1 or TSC2 mutations. The fourth type, which is rare, but definitely an association, is what he called microcystic disease. And in this form of the kidney, of cystic disease in the kidneys in tuberous sclerosis, there are very, very large numbers of very, very tiny cysts. And he warned that this type of kidney problem could be missed on an ultrasound scan. It might be detected, but couldn't be detected in a robust way. And he said it was better seen by MRI scanning. And it was important to recognize this microcystic disease because it can lead to kidney failure. And then the final type, if I got it right, because it was quite complicated, the final type of kidney disease or cystic problems that he described were what he called focal cystic disease, where a particular area of a kidney developed cysts following some sort of damage. So the kidney would be normal, and then this cystic area would appear quite rapidly. And he felt that that was very often after the kidney had been damaged. For instance, if someone has a very prolonged seizure, the kidney and the other tissues can be starved of oxygen, and the blood flow to the kidney can be changed dramatically, and that can lead to damage to the kidney tissue, which then leads to cystic changes in the kidney. And again, this sort of change can be associated with a sudden reduction in kidney function at the time that the damage occurs, and then a bit of recovery, but not complete recovery. And so this is important to recognize as well. So that was the first bit of John's talk, and he gave, he put up very nice pictures of the scans of patients with these different types of cysts in the kidneys to help us to understand how to distinguish between them. And I guess the importance was that, some of these cyst problems were associated with a high risk of kidney failure, and others with only a very low risk of kidney failure. So quite important to try and distinguish these. And he then went on to tell us something very interesting about his work to try and understand how cysts develop in tuberous sclerosis. And what he described was that the cells inside cysts, which are really just fluid-filled spaces in the kidney, and those fluid-filled spaces are lined by cells. And he said that a small number of those cells in any cyst are so-called TSC null cells, where both copies of the tuberous sclerosis gene are mutated and don't work. And so these would be sort of, if you like, genetically classical TS cells. And he said that these cells in kidneys secrete little particles called vesicles. They're little membrane-bound particles that are secreted into the cyst. And what it seems from his research is that these vesicles are then taken up by other cells in the proximity of these TSC minus cells, and they incorporate some substance from these vesicles that changes the appearance and the behavior of cells that would previously have looked more normal, and makes them more abnormal. So this was I think quite a new thought in relation to tuberous sclerosis, the idea that a cell that is essentially causing the tuberous sclerosis related manifestation can affect other cells that are in its vicinity by secreting these vesicles. And he treated these cells that were secreting vesicles with mTOR inhibitors and found that this reduced the production of these vesicles and reduced their effects on surrounding cells. Now, what this, I think, really raised was the question, well, what is in these vesicles that these cells are producing? And if these vesicles contain some sort of molecule or chemical that's produced by these abnormal cells that promote cyst growth, can we identify that substance and a treatment to block it? And as I say, it seems like rapamycin or mTOR inhibitors perhaps do block these vesicles, but of course they block all sorts of things being synthesized. So perhaps a more specific treatment could be found if there was a better understanding of the role of these vesicles in causing kidney cysts. So I think this is quite new and sounds exciting, but I think it's still at quite an early stage. So that was kidneys for us. Any questions on kidney cysts?
So not so far, but
sorry? It was quite complicated, wasn't it?
Yes, it was. It started off, it was the first bit was beautiful because he had all those beautiful illustrations of the cysts, and then it got more complicated. But I think, just as a general message, actually, I think the cystic kidney disease in TSC is quite poorly understood by non-experts. And I've certainly had I work with a wonderful nephrologist, pediatric nephrologist, who very much categorized the renal cystic disease in TSC as being related to PKD, and didn't really recognize that there were different types. So he kept questioning me about what's the genetic basis of this child's TSC. It must be PKD because they've got cysts. And I kept saying, no, it doesn't have to be PKD. So I think that actually, you know, this is not as well recognized as it could be. So Adrian is asking, would your average consultant nephrologist be able to determine between these cysts? And I think actually that question probably is more towards the radiologist than the nephrologist, although many nephrologists are very good at interpreting scans. But the radiologist would usually be interpreting the imaging. And it's a very good question. I think, yes, it would depend on, you know, the location of the cysts, but there may be, I think, some difficulty in differentiating between the sort of classic PKD and the multicystic kidney disease, which I think is where I've had my interactions with a nephrologist. Julian, do you have any particular thoughts on that?
Yeah, I think it is pretty difficult. You know, I think people like John Bisler, who, you know, spends hours and hours, days and days looking at these scans and trying to think, well, is this what's going on here different to what's going on there? Whereas I think on the whole, you know, when you get a scan done, the report will usually come back saying, you know, there are multiple cysts or something quite unhelpful like that. And what I often find is a good thing to do is to go and discuss the scans with the radiologist, and to ask for more, or at least to send the report back and say, well, you know, how many cysts, how big, whereabouts in the kidney, is it in one kidney or is it in both kidneys? And really, you know, I think patients, again, should ask these questions, should ask for more detail, because really just being told that there's multiple kidney cysts is not enough. You need a much more detailed interpretation of the scan than that. So does that help, Adrian? I can see Adrian there, if it's the same Adrian.
Thank you. He says yes. Thank you.
But you know, this is I think this is quite at the sort of cutting edge of thinking about this cystic disease, that it, you know, that there are so many different types with these different outlooks. Yes,
I agree.
I would say send your scans to John Bisler, because he's always very helpful. But obviously, he can't have every patient in the world's scan to look at. But yeah, my experience is that he's very interested and will usually do what he can.
Yes, and I think with him, you know, increasing our knowledge, I think our radiologists and our nephrologists will become more familiar with this. Actually, it's about a, you know, sort of matter of us disseminating this out. And you know, we have a tuberous sclerosis MDT, and we have an outstanding renal radiologist who helps us, and he's now trained up a new, she's a registrar who's just sort of come through, and a young consultant, and so she's learning from him. So we're actually really lucky at St George's, because he does recognise the difference. So I don't, you know, maybe not all the subtleties that John recognises. Well, we will see. Anyway, I'm conscious of time, and I know that the last set of talks will be of great interest to lots of people. So let's move on to the TAND and autism cluster, as it were.
Yeah, so we had some interesting talks, and so I've put these ones together because they were all quite closely related. So there were basically three talks, the third of them with two different speakers. So Mustafa Sahin is a pediatric neurologist from Harvard Medical School in Boston in the US, and he told us about some studies that are going on in the US under a sort of an umbrella term that, for their study called, is the TACERN collaboration. All the researchers love these sort of little, these names for their studies. Charlotte Tai, who's based at King's, the Institute of Psychology, Psychiatry and Neuroscience in London, gave us an update on the EDIT study, which some families in the UK might have been involved in. And then Petrus de Vries from Cape Town and Anna Janssen from Brussels told us about the TANDEM study. So it was a real international, set of talks. So first of all, I'll just give a little bit of information about what was reported by Mustafa Sahin in relation to the TACERN study or collaboration. And this is a US-based study in 5 of the really big children's hospitals with large tuberous sclerosis clinics. And what they're doing is basically they're trying to recruit as many young infants with tuberous sclerosis aged between 3 and 9 months of age as possible. They've got up to 166 babies have been recruited so far. And they're studying them really quite intensively with MRI scans, including diffuse and tensor imaging, which is a sort of ultra detailed way of looking at pathways in the brain, by EEG, the brainwave test, the electrical brainwave test, where you stick electrodes on the scalp and measure the electrical activity in the brain, by neuropsychological tests, which I think are pretty basic in children aged 3 to 9 months, because obviously there's a limited range of things that they can do. But these are really sort of developmental tests to see how the child's development is progressing. And they're also taking blood tests. So this is pretty onerous from the point of view of the babies and their parents. And what they were really interested in looking for were there early signs in these tests that could indicate a high risk of autism or developmental problems, and what were the effects of treatments, different treatments and different timing of treatments on the emergence of autism as these infants got older. And he described their initial findings, first of all, that the EEG on average showed abnormalities about 2.5 months before clinical seizures became apparent. So this is a finding quite like the finding from the EPISTOP study in Europe and the US that's run by Sergiusz Josiek from Warsaw in Poland. So both the TACERN study and that EPISTOP study have found that if you monitor the EEG regularly during infancy, you can detect changes occurring in the brainwaves before seizures develop. And if treatment is given with an anti-epileptic drug, particularly vigabatrin, when the EEG becomes abnormal, but before seizures develop, there seems to be a better developmental outcome for the baby with tuberous sclerosis. What the TACERN study also showed was that when vigabatrin was given at a higher dose, over 150 milligrams per kilogram body weight per day, as opposed to a lower dose, there seemed to be a lower risk of seizures relapsing. And of course, often seizure relapse is associated with developmental problems, with a change in acquisition of new skills by babies. So I think the results from the TACERN study so far, I think, are broadly supportive of the recently published results from the European EPISTOP study, and suggest that in future there will be a place for monitoring the EEG of newborn babies with tuberous sclerosis and intervening with an anti-epileptic drug before seizures actually develop. So that was interesting but this will be a long-term study, and I guess these were really just the earliest findings. Charlotte Tai told us about the EDIT study. This has been running for quite a few years now, again supported by the Tuberous Sclerosis Association. This is a UK study, and it's trying to understand better the development of babies affected by tuberous sclerosis. And so what Charlotte and her co-workers have been doing is they've been studying these babies really quite intensively largely through visits to the family home to observe how the babies are developing. And they've recruited so far 32 babies with tuberous sclerosis and 28 babies as controls who do not have tuberous sclerosis, who are going through the same developmental assessments. And what they have found is that the development between the babies with TS and the control group starts to diverge, it seems, typically at around 5 months of age. And thereafter, the average baby affected with tuberous sclerosis doesn't catch up so the development starts to diverge and then stays behind from about 5 months of age. And one of the facets that seems to be very important in determining this developmental trajectory seems to be the onset of seizures and the control or failure to control the seizures during this first year of life. So I think the group have been funded now to follow this group of children up through to school age, and clearly more information will emerge over the coming years to show how seizures and the treatment of seizures is impacting on development over those important first years of life. But I think the main message for me was, yet again, the earlier seizures can be controlled or prevented, the better in tuberous sclerosis. So the third talk in relation to these problems, TAND or tuberous sclerosis associated neuropsychiatric problems, is the TANDEM study. So Petrus de Vries and Annie Janssen are leading this study, which is really a global one. And it's part research study and part an attempt to empower, I think that's what the M stands for, empower families to really have these TAND problems properly communicated to medical and paramedical professionals so that they can be properly addressed. And to do this, the group have developed an app for a mobile phone that enables systematic data entry, from day to day, so that when families go along to clinic and want to describe the TAND problems, the behavioural developmental problems that children are experiencing, this is being recorded in a way that will make sense to the professional and will enable comparison with expectations. So hopefully the app will result in an improvement in the quality of consultations, because often, perhaps not so much in tuberous sclerosis clinics, but in many settings, of course, consultations are rushed and the professional may not be an expert, as most of us are not, in developmental or psychological problems or behavioural problems. So this will be, hopefully empower families to have information about these problems presented in a coherent and meaningful way to professionals that they can understand. And at the same time, the hope is that the use of this standardized app by many TS patients and their families will result in a large database of standardized data on these problems, which will improve our understanding of them. But this project, as far as I could tell from the talk, was still at a pretty early stage of development. So they've developed the app, and my understanding is that they're validating it at present, making sure that the data collection and quality is consistent across, not just across children of different ages, but also across geographies, because the study involved Asia, for instance. Families in India were being recruited, from Australasia, from South Africa, from Europe, North America, and so on. So clearly there were a lot of things to validate, not least of them differences in language and ways that these problems are described. So I think, was that it? Yep. For the TAND type problems.
Thank you, Julian. Sorry, I was muted for a moment. So no questions just yet, but do get them in if you have any. And so there's just a question about the going back to the cysts and the kidneys. Can I request scans to be sent to my TSC clinic, or should I speak to the clinic first? My child just had a kidney ultrasound. So I would say, first of all, it depends a bit on the age of the child, whether just an ultrasound is enough. If you would like a second opinion, I would always speak to your clinician. Okay, 11. I would always speak to your clinician and ask about the interaction with the local TSC clinic. But also it will depend slightly on the results of the scan. So if the scan is completely and utterly normal at the age of 11, there's probably not a huge amount extra that the TSC clinic could offer. But if there are some abnormalities on there, then it may well be worth running it by a specialist opinion to decide whether more and different imaging is indicated, because we would always say, that at some point a baseline kidney MRI should be done, but it would depend on the results of the ultrasound. So that would be my view, but I would always confer with your clinician who can facilitate that for you. So we are getting on to some questions about the TAND. So would the app be just for children or also for young adults with TSC? So my understanding is that this is applicable to young adults as well. So children and young adults. So it's effectively, it's an electronic version of the TAND form. I can't remember exactly what he calls it, but
it's a checklist, I think.
Yeah, checklist, TAND checklist that Petrus de Vries and his team developed. So they've basically put that into an app form, and certainly in our clinic, I have to put my hands up. We don't always have time to go through TAND, but this we would use it in both children and young adults. Okay, so there's some questions that I'm going to struggle to answer. So first of all, when is the app expected to be available? I don't think we were given a time, were we, Julian?
No, because all they said was that they were currently involved in validating it. So clearly some people must be using it in order to validate it. But I think, in a research study setting at the moment, rather than being generally available.
Yes. Okay.
I think some centres are in the UK are involved in this, but our centre in Cardiff hasn't been involved in the TANDEM project. I don't know if anyone. I know there are European centres involved, that's for sure, but I don't know if any patients or families in on the call have been involved in the research phase of this that could tell us.
We haven't in St George's, but there may be that other centres have been. So if you have been, put it in the chat. So this is the one that I'm struggling with a bit. Celine asks about an 18-year-old patient. Is there any data about conversion disorder in TSC or other psychiatric disorders? So specifically conversion disorder, I don't have any information or knowledge of that specifically in TSC. Certainly, if you look at psychiatric disorders in the round, yes, there's a lot of data about psychiatric disorder, and Petrus de Vries has done a huge amount of work in addition to Patrick Bolton in the UK, and I'm sure there are other psychiatrists around the world who've done other work. Julian, conversion disorder in TSC, do you have any experience?
Yeah, I don't have any specific experience of conversion disorder in TS. I don't know whether the family that have posed this question are finding problems with accessing appropriate expertise, which is a huge problem for the psychiatric and psychological manifestations of tuberous sclerosis. But if you are experiencing difficulty, then I think the best thing to do is to ask to get to your local tuberous sclerosis specialist clinic and to seek onward referral for psychological or psychiatric help or assessment from there. But I have to say, in Wales, we really struggle with appropriate services for this, and the waiting lists are really long. That's one of the, I think the services are flooded, and so it is an area where undoubtedly more resource is needed.
Yes, I totally agree. I think that's almost universal. And so Peter's asking, hi there, regarding TANDEM first year seizure control, does this mean if the child suffered seizures, if the child suffered seizures for a few months before getting them under control, the likelihood is any developmental delay will remain through their life? So it's quite a difficult question to answer, but there is, in general, we know that early, as Julian has alluded to in the talks, that early control of seizures tends to be associated with a better outcome. But again, we're talking about groups of patients, so we're always looking about across patient groups, and so when you come down to any one individual patient, it's hard to generalize. So the general trend would be the better and earlier the seizure control, or the later the seizures occur, then the likelihood of typically developing child is going to be increased, and the reverse also holds. But it's difficult to say in one particular child that an outcome is necessarily going to be good or bad for that child. Julian, have you got anything to add to that?
Yeah, I think it's, as you say, it's risk, isn't it? So the risk of having developmental delay or developmental problems is higher if the seizures occur early or are not controlled quickly. But it doesn't mean it will happen, but the risk is increased. And the other thing is, I don't think it's ever too late to strive for better seizure control. So I think, I've certainly seen patients in my clinics where I felt that, the seizures, despite perhaps the patient being on one or several anticonvulsants, the seizures are still not adequately controlled, and it is worth really trying to get the seizures under control, and a paediatric neurologist needs to be involved, preferably a paediatric neurologist with a special interest in tuberous sclerosis. So I think that should be the aim. But of course, for many young people with tuberous sclerosis, the seizure control will never be complete. But the better it is, the better the chance of good outlook. So I think it's just always worth trying to improve seizure control if it is inadequate.
Absolutely. Okay, so we don't have any more questions. So if people do think of questions afterwards, do send them across to Luke, and either I or Julian will do our very best to answer them. And I'd just like to thank everybody for coming today. It's been really fantastic to have so many people listening in to this. And I particularly would like to thank Julian, because he really did do almost all the hard work. So that's very, very kind of him. And thank you to the TSA for organising the session.
Yeah, and just to reiterate the thank yous, a big thank you to you as well, Frances, and to you, Julian, as well. And of course, thank you to everyone for joining as well. Some absolutely brilliant questions, which I've been noting down furiously on my notepad. I think I've got them all. So if you want to look at this again, we will be sharing it on our website and social media, as I said at the beginning. And yeah, don't forget about other events that we'll be holding. Keep an eye out. And just before I let you all go, although you can go whenever you want, I guess, just a reminder that the TSA is always here for you, especially during this hard time. The TSA has gone through changes this year as well, but the bottom line is that we're still here to provide help for today and the queue for tomorrow. With that, I'll let you all go and just say thank you again. So have a good night, everyone. Bye.
Bye, everybody. Bye.
Bye for now.